In this video, we can discuss about the scope or importance and applications of microbiology. It's an important topic of pharmaceutical microbiology of third semester B-Pharm. It's a frequently asked question: what is the scope of microbiology and history of microbiology? Now coming to the content. In this video, we will discuss the scope of microbiology in different fields like medical microbiology, public health microbiology, ecological microbiology, agricultural microbiology, food and dairy microbiology, industrial microbiology, microbial physiology and biochemistry, microbial molecular biology, microbial genetics, and pharmaceutical microbiology in detail. Now coming to the scope, microbiology has great impact on pharmacy, medicine, agriculture, food science, ecology, genetics, biochemistry, molecular biology, recombinant DNA technology, and genetic engineering. Now first one is medical microbiology. As I mentioned in the branch of microbiology, the medical microbiology, in which we will deal with the identification and isolation of infectious bacteria and the planning method and measures for the treatment of and the elimination of that infectious bacteria and tracking down of new undefined pathogens, and by discovering their mechanism by which they will cause the diseases. This is the first scope, medical microbiology. Now second one is public health microbiology. Microbiology is used to measure and to prevent the spread of communicable diseases, and also it deals with the monitoring of community food establishment as well as water supply to keep them free from the microorganism. Now third one is agricultural microbiology. Now we are living in a era of we are using organic vegetables. So microbiology has a large impact on agriculture. In this branch has developed method to combat the plant diseases that attack the food crops and to increase the soil fertility and crop yield. It also includes the study of microbial flora in the digestive tract of various animals or organisms, and bacteria and virus have been developed as an insect pathogens which can substitute for the chemical pesticides. So microbiology is used in the field of agriculture for soil fertilization and to yield excess of crop. Fourth one is ecological microbiology. In this we will study about the contribution of microorganism to the carbon, nitrogen and sulfur cycle in soil as well as fresh water. This also study on how microorganisms can be used to reduce the pollution effect of environment. Now fifth one is microbial physiology and biochemistry. In this we will study the pathway of the synthesis of antibiotics and toxins by different microorganisms and microbial energy production and way in which the microorganism can survive the harsh environmental condition and the effect of chemical and physiological agents on microbial growth and its survivals. So that is the fifth scope, microbial physiology and biochemistry. Sixth one is food and dairy microbiology. Microorganism is the one of the key organism or key component for the production of different food products like cheese, yogurt, pickles and beer. This stream also identify and prevent the food poisoning by botulinum as well as salmonellosis. Now, microorganism is used for the treatment of industrial waste, usually used to decompose such waste like water, salt, organic matter and spent media, which may be toxic, by different microorganisms like actinomycetes and fungi and protozoa. Sometimes anaerobic bacteria in anaerobic digestion tank are active in the decomposition of organic matter by yielding simple organic molecules like acid, alcohol and glycerol and amines. So in anaerobic culture, microorganism can be used for the production of alcohol. In cyanide, which is produced by industrial scale use of metal extraction and electroplating as well as steel industry, which can be degraded by Streptomyces labyrinthidae. So these are the scope in treatment of industrial waste material. Next one is microbial molecular biology. In this, we will use microorganism for the studies, the nature of genetics, information and how it regulates the development and function of cell and organism. Now, ninth one is microbial genetics. It deals with the producing a new microbial strain that are more efficient in the synthesis of new products. So sometimes we will produce a new strain of microorganism which have a capacity for the production of high yield of an fermented product, or sometimes this fermented product may cause inhibiting action on the same microorganism. That time we will produce a new strain with antibiotic resistant strains of that microorganism can be produced by using microbial genetics. And 10th one is pharmaceutical microbiology. In pharmaceutical field, microbiology is used for the many purpose. We will discuss in detail one by one. First, pharmaceutical microbiological scope is for the performance or for performing antimicrobial assay of antibiotics. So, any other drug other than antimicrobial agents we can study in animals. For example, the drugs which is used as an antihypertensive, that is used to decrease the blood pressure, we can study the action of that type of microorganism in any laboratory animals like rabbit. For that we have to inject some drugs which will increases the blood pressure. After that we will inject our drug which has to be studied for their action. So after injecting the drug or new drug, if the BP of that animal is reducing, we can find out that the effect of this drug is antihypertensive. But except antimicrobial agents we cannot study in large animals. We have to select one microorganism. So antibiotics capacity to inhibit the growth of microorganism has to be studied using antimicrobial assay. This can be done by different methods like chemical, enzyme based, turbidimetric and or biological agar fusion method. By performing the antimicrobial assay, we can find out the minimum inhibitory concentration of an antibiotic or antimicrobial agents. What is mean by that? So if we are giving more concentration of antimicrobial agent, it may be kill the microorganism, but also it will cause some problems to the host cell also. So So if we are treating a infectious condition in human with an antibiotic with high dose of antimicrobial agents, it may kill the microorganism, but also it will cause some harmful effect to human. So for that we have to find out the minimum inhibitory concentration of the antibiotic, that at that concentration all the microorganism will be killed and it will not cause any harmful effect to the human being. So to find out the minimum inhibitory concentration also, we have to perform antimicrobial assay of antibiotics. So how we are doing this antimicrobial assay? We have to prepare one sterilized culture media. Culture media is nothing but a media or nutrient composition that will help for the growth of microorganism. And after that we will inoculate means we will transfer a suspension of microorganism to the fresh sterilized culture media to get a uniform growth of microorganism on that culture media. After that we will place antibiotics discs which we have to study on the surface of agar plate. And after incubation means we have to give the optimum conditions for the microorganism to grow. So after a minimum of 24 hours, if we are observing the culture media, we can see a zone of inhibition. That means, see here you can see the zone of inhibition. That means at that area, due to the effect of antibiotic, this antibiotic is diffused through the agar media, will cause the killing of microorganism in that area. So here one antibiotic disc is there which is not kill the microorganism, which indicate that antibiotic is resistant to that microorganism, and this is another antibiotic which have a small zone of inhibition, diameter of zone of inhibition, which indicate which have some power to kill the microorganism. See, this one is another antibiotic disc which have a large zone of diameter of zone of inhibition, so it have more effect. So by this method we can find out the minimum inhibitory concentration. For example, if we are considering from a lower concentration to higher concentration, so in which lowest concentration the growth of microorganism will be killed, that is the minimum inhibitory concentration. Now this is the turbidimetric method. So agar plate method is an microbial assay which is performed on solid culture media. This medium will be solid. Now turbidimetric method is a method of antimicrobial assay in which we will use liquid media. So in liquid media, if there is any growth of microorganism, the turbidity nature of culture media will be produced. So by observing the turbidity in the different tubes in which we have taken the growth, we have taken the different concentration of antibiotics, we can see a concentration in which the growth of microorganism will be inhibited. So this is 0% of an antimicrobial agent in which the turbidity is there. As the concentration increases, the growth of microorganism will decreases, and at a point there will not be any growth at a concentration. So the concentration of the or this is the concentration, lowest concentration of antibiotic in which the growth of microorganism will be inhibited. So this is another pictorial representation. This is turbid. This is also turbid. So by increasing concentration 2 microgram per ml and 4 microgram per ml, on coming to the 8 microgram per ml, in which the growth of microorganism is inhibited, as we can observe as a clear solution. So this is the minimum inhibitory concentration. This is also giving a clear solution which will inhibit the growth of microorganism, but the problem is, by the definition of minimum inhibitory concentration, it is the lowest concentration of antimicrobial agents at which the growth of microorganism will be inhibited. Now coming to the next scope of pharmaceutical microbiology, that is the evaluation of disinfectant. We know the disinfectants are the agents which are chemical agents which are used to kill the microorganism on inanimate objects. So specific strains of microorganism are used for the evaluation test, like phenol coefficient test, suspension test, capacity test. So these are the different tests for the evaluation of disinfectant. Now coming to the next scope of pharmaceutical microbiology, that is efficacy testing of preservative. So preservatives are the agents or antimicrobial agent which are added to different products like food products or different pharmaceutical products to prevent the growth of microorganisms. So we have to evaluate the efficacy of a preservative. So that can be done by using various strains of microorganism. In this test, separate containers of the product are inoculated with a known concentration of variety of test organism, and over a period of time samples are removed from each container and tested for the proportion of survivors. Now coming to the next scope, that is the bacterial endotoxin test. It is involved in the quality control of parenteral or injections and the medical devices. It can be done by Lalter's gel clot methods, chromogenic endpoint method, kinetic turbidimetric assay method, or kinetic chromogenic assay method are used to find out the bacterial endotoxins. Next one is for the evaluation of bioburden in production area. So production area of pharmaceutical products, especially the parenteral products, which are the products we will inject directly into the bloodstream. So it should confirm that there is no presence of microorganism. So in order to evaluate the bioburden or the presence of microorganism in the production area, we have to perform an settle plate method. Settle plate method is nothing but we will prepare a fresh culture media, solid culture media in a petri dish, and we will open it for 1 to 4 hours in different area of production, and we will incubate and we will observe for the bacterial growth. So after the incubation, if there is any growth like this picture, this is some colonies of microorganism. So if there is any colony in the plate, it indicates that the presence of microorganism in the production area. So we have to sterilize that one. We have to disinfect that production area to control the microorganism and for a better product. Now coming to next scope of pharmaceutical microbiology, that is microorganism is used as a biological indicator to evaluate the efficacy of sterilization process. We are performing the sterilization process in a belief that all the organism will be killed by this sterilization process. In order to assure that, we will use some microorganism which have an higher resistance to that sterilization process. After that sterilization process, if that organism is survived, that indicate the failure of the sterilization process. So see, these are some examples of microorganism which is used as an indicator. In case of moist heat sterilization, we will use spores of Bacillus stereothermophilus, and in case of dry heat sterilization, that is hot air oven, we will use Bacillus stereothermophilus and Clostridium tetani. And in case of ionization radiation sterilization, we will use Bacillus spumilis, and ethylene oxide sterilization we will use Bacillus subtilis and Aspergillus. Next next scope of microbiology in the field of pharmaceutical microbiology is detection of presence of microorganism in pharmaceutical dosage form. So every pharmaceutical dosage form are prepared in an intention to treat against some diseases. So it should not contain any microorganism. Especially some microorganism will be there in some pharmaceutical products like topical products like ointments, which containing oily natured oils. And as per the pharmacopoeia, USP has described the detection test for E. coli, Staphylococcus aureus, Pseudomonas aeruginosa and Salmonella, which must be totally absent from the preparation, which are hazardous organisms. So these are some examples of microorganisms that should be absent in different products. For example, topical non-sterile respiratory products like inhalers, nebulizers, it should not contain Pseudomonas aeruginosa and Staphylococcus aureus. And the different pharmaceutical products which are used in the route of oral as well as rectal, like suppositories, it should not containing E. coli, and oral products which containing natural origin materials, Salmonella, E. coli, Staphylococcus aureus should be absent, and in herbal remedies, Salmonella and E. coli should be absent. Now, last scope of microbiology is for the production of or manufacturing of different pharmaceutical products like antibiotic. Many of the antibiotics are produced by fermentation process by using different microorganisms. Examples are penicillin, streptomycin, tetracycline. These are the different antibiotics which are produced by fermentation process. And some vitamins like vitamin B12 also produced by fermentation process by using microorganism. And for the production of intracellular and extracellular enzymes like amylase, protease, lipase, invertase, oxidase, catalase, cortisone reductase, phospholipase, cellulase and chitinase are some examples which are also produced by fermentation process by using microorganism. Now, different hormones like humulin, which is a human insulin produced by fermentation, as well as recombinant DNA technology. In that also we are using the microbiology, and for the production of different steroid like cortisone, prednisolone, ergosterol, testosterone and progesterone. These are produced by fermentation process by using different microorganism. And for the production of vaccine like poliomyelitis, BCG, DPT, tetanus toxoid, all these are produced by using microorganism or its product. And last one is some organic compound like butanol and ethanol and acetone are produced by different microorganism by fermentation process. So these are the different scope of microbiology. So we had discussed about medical, public health, ecological, agricultural, food and dairy, industrial, microbial physiology and biochemistry, microbial molecular biology, microbial genetics, and finally pharmaceutical microbiology in detail. History and scope of microbiology as a 10-mark question, and scope and application of microbiology as 5-mark question, and importance of microorganism in the pharmaceutical field. So that is all about scope of microbiology. Thank you for watching this video.